I'm going to give a brief introduction on the status of Philippine orchid systematics in this paper. I'll proceed on to presentation of methods and results of our research on DNA barcoding of Philippine plants, but for now, for here, I will be presenting those results for orchids. The research is actually done in order to narrow down our selection of the best DNA barcode that would apply for orchids. And I may be discussing in passing our other initiatives or other initiatives that we have on the Philippine flora. So here in the Philippines, we have a very rich orchid flora. We have about 1,142 species under 152 genera. And if there are about 8,000 species of orchids here in the Philippines, then that should represent about 10 to 15% of the Philippine flora. So if we are able to barcode a large sample of orchids, then we should be covering a wide base for the Philippine flora. The Philippine orchid flora also has a very high endemicity. That's about 90% at least. So compared to other countries in Southeast Asia or in the Malaysian region, including, let's say, adding into it Vietnam from the Indochina region, the flora of the Philippines, the orchid flora of the Philippines is comparatively or relatively high. No, among our most species genera are Bulbophyllum. There are 135 species at least. They are mostly found in highland or montane environments. They're difficult to collect and difficult to grow in the lowlands, of course. So if we need to barcode them, then they go directly to the lab. Dendrochilums are well studied. It has been revised some years ago. There are 90 species. Most of them are ornamental. The Dendrobium are very ornamental, collected by a lot of hobbyists in the Philippines and abroad. There are about at least 80 species. The Urea species, well, are quite problematic. They are there are about 61 species of Ureas in the Philippines. And in our collection of the DNA, we have, these are represented, there are different genera also represented in our collection. In the past 10 years, about 54 new species have been discovered under 25 genera. In fact, there are at least 3 monotypic genera which have been discovered during this period. So this is just to show you the interest that goes into orchid systematics here in the Philippines. And we can see that over the decade, much of these discoveries came around 2005 to 2010. Perhaps that is because a lot of taxonomists are now are able to do systematic research. They are able to publish in international journals for their species discoveries, and there have been a lot of collections through the Flora of the Philippines project, which also contributed to the data available for taxonomists. So in those past 10 years, the discoveries for the entire Philippine flora mainly came from the orchid group. About 20%, 28% of that came from orchids. Of course, there were new species from one big group like Rubiaceae, mainly because there was one genus which was a big genus which was revised during that period. But most of those species like orchids that have been described in the past years were based on traditional methods, being able to distinguish differences based on morphology, anatomy, and palynology. So there only have been few papers or few discoveries which used molecular evidences, and that is because probably we have there are limited resources in this case. So this research enabled us to do more taxonomic research by incorporating DNA barcoding into the as basis for our research. So what we do is we collect orchids from the wild. We also go to local communities, ask for samples from backyard gardens. We go to botanical gardens around the Philippines. We even buy specimens from local markets, from Sunday organic markets, and we frequently go to the field as well to collect uh, samples. So if possible, we keep a living collection, but mainly we keep the specimens, or most of the specimens that we study also go as vouchers. So we apply the usual method following the protocol that Dr. Kress gave us some years back for DNA barcoding. We keep our samples in the university, just within the university. We try to be very systematic about it. We keep them in low temperature freezers, and going around Manila or the country, at least we have found our method to be more or less the same with one other major research institution in the country. So far we have barcoded at least 100 orchid species. And if we have to gauge on which is the best barcode, since we only barcoded we only use 3 sequences, the MAT-K, the PSBA, and RBCL. Based on the robustness and PCR amplification and sequence alignment, we found MAT-K and PSBA the easiest to amplify, though we found some species, taxa, to be difficult in terms of amplification or even extraction, like that of Paphiopedilum and some members of the Epidendroideae group. In terms of amplified gene length, RBCL is the shortest, which is 774 Ts, MAT-K about 800 base pairs, and PSBA also about 800 base pairs. In terms of the gene being able to identify or define interspecific sequence differences, we found the RBCL to be the best with the highest value, followed by MAT-K, and then the lowest was found for PSBA. In terms of ability to identify or define intraspecific sequence differences, the best was for RBCL, followed by PSBA and MAT-K. RBCL with the least value for divergence within species. Therefore, the best to identify or to act as barcode. In terms of nucleotide diversity or divergence, again, the best was the RBCL with the highest value, followed by MAT-K, and then the least is PSBA. But we use another criterion based on a previous study by Mayer and colleagues on Diptera. We try to compute for pairwise distance thresholds. We try to measure genetic distances. As explained by John earlier, if there are overlaps in the genetic distances, then those barcode sequences or those sequences may not be that useful. So we did that again for determining the uniqueness of the DNA barcode to test whether identical sequences are shared by individuals from different species as well. So computing for that, we found that the RBCL could be the best or most reliable DNA barcode with the interspecific sequences and the intraspecific sequences defined by a wide gap of about 6 to 7%. For MATK, there seems to be little overlap there between the sequences. However, for the tRNH and PSBA, there is a very great overlap in the sequences. Thus, this is the least reliable barcode in our study. And the best, of course, was RBCL. So, the best barcode should then be RBCL, which has the shortest gene, the greatest ability of species differentiation, which exhibits also the highest nucleotide diversity, and MATK as well. Well, based on other literature, MATK is also very good for orchids because it's the most easily amplified in sequence, effective in determining interspecific differences, and with high nucleotide diversity. We're using also the DNA barcoding technique to study problematic genera or groups, like for example, Oberonia liparius and Stichorchis. The rhea complex, which we are studying now, whereby most of the species in the past are identified or classified as rhea, but now there has been a lot of movement. Some of the species are moved to Micaranthus, Spinalia, Trichotoscia, Sideria, and Coelenterobus. And we're also looking at the differences of individuals of new genera like Sadocum, which came essentially from Grammatophyllum. So aside from orchids, we're also using the technique to identify, to differentiate individuals within certain genera like Colocasia and Alocasia in aroids, in the Araceae family. We're looking also at different species of cinnamon. Cinnamon species are very difficult to identify, especially because they're so diverse, the flowers are so small, and these are highly economical and highly economic for its medicinal value here in the Philippines. So it's really essential to identify this species correctly. Another group of plants where we are applying DNA barcoding is on cycads. We have established identity of certain species or new species of cycads based on DNA barcodes, which otherwise are difficult to identify, mainly based on morphological characters. We're incorporating also our data in a database which runs in the BRAM system, which is commonly used in Malaysian herbaria like the Philippine National Herbarium, Singapore, as well as the National Herbarium of the Netherlands. So here we put in specimen information, living collection, we incorporate digital images, and of course link up to the GenBank for the sequences. We would like to exchange also material, and if ever we go on to that, we would like to take up the model of the Q material supply agreement for exchange or sharing of materials. So for this research, I'd like to acknowledge the Commission on Higher Education, which funded our research, our university, the Philippine National Herbarium, where we deposit mainly our collections, and taxonomists, orchid taxonomists like Eddie Vogel, Andre Schittemann, Jaap Vermeulen, Henrik Peterson, and Jim Kootz, who help us identify our orchid collections. I think that's it. Thank you.
Thank you, Esperanza. I didn't get to unmute in time for you to hear the round of applause you got in the room. What a lovely talk. Questions for Esperanza, please enter them online, and I'll use the chair's prerogative to ask some questions. First, Esperanza, where did you do the sequencing? Was this all done in country?
No, we can only do up to PCR, and then we send our samples to Malaysia. But previously we send our samples to Korea. So either Korea or Malaysia, because it's cheaper to do it that way as well.
Okay. Is Malaysia a commercial sequencing company or another
it's
academic
commercial
lab?
It's a commercial entity. It's a commercial lab.
I was also surprised at the results that RBCL was so good at separating species. It's generally considered slower than MAC-K. Is that because this was a floristic treatment where you had were the species not that closely related?